Yeah, let's welcome Dr. An Jie, Secretary General of Seattle Chinese Biomed Association (CBA), to introduce Dr. Zheng Ning.
Hello, everyone. Can you hear me?
Yes.
Yes.
Okay, thanks. Hello, everyone. This is Jian. It's my great pleasure to introduce Dr. Ning Zheng, the former president and current board member of our Seattle Chinese Biomedical Association (CBA). Dr. Ning Zheng is a professor at the Department of Pharmacology in the University of Washington School of Medicine. He is also a Harvard Hughes Medical Institute investigator. Ning obtained his Ph.D. in 1997 from University of Texas Southwestern Medical Center at Dallas, and was trained as a structural biologist at Memorial Sloan Kettering Cancer Center in New York. After joining the UW faculty, he has established a highly productive research laboratory studying the molecular and structural mechanism by which protein-protein interactions control basic biology and human diseases. His research program spans many areas of biology. His lab has published many high-impact papers, including more than 20 publications in the three most prestigious scientific journals, Nature, Science, and Cell. Based on the funding made by his lab, he has co-founded two startup companies, which are actively developing novel small molecule drugs for major human diseases, such as cancer and neurodegenerative disorders. Congratulations to Dr. Ning Zheng for this 2020 CIE Asian American Luminary Award, and also for being elected to Washington State Academy of Sciences in 2020. Welcome Dr. Zheng to share his excellent work now. Thank you.
Thank you, An Jie. Let's welcome Dr. Zheng Ning.
All right, so I will start to share my screen. I hope everybody can see my first slide.
Yes.
All right. Okay, thank you, Anjay, for the very generous introduction. And it's really a great pleasure to be here to speaking to you today. Before I start, I would like to thank the Seattle Chinese Biomedical Association for their nomination, and of course the CIE Seattle chapter for this prestigious aluminum award. I'm really honored and humbled by this award, and consider it really as a high recognition of the achievement made by many talented and dedicated students and trainees that I have been fortunate to work with in the past 2 decades here at the University of Washington. Nothing was possible without their original and relentless work. Meanwhile, I actually also want to thank you all, the audience, for spending this really fine Saturday morning and tuning into a presentation that's perhaps distant to the expertise of most of you. And to honor your precious time and interest, I decided to spend the next 20-30 minutes also to give you a rather bird's eye view of what our research is all about and what we are most excited about these days in addressing the increasing challenges we're facing in biomedicine, and particularly the development of novel therapeutics. And I hope my talk will be both informative and entertaining. So we all know that the past year was highlighted by COVID-19 pandemic, which hopefully will be tamed soon, thanks to the availability of vaccine. I'm also excited that Dr. Shanli Liu will speak about this topic in the second half of this hour. Meanwhile, let's not forget that the modern society has been facing and will continue facing numerous major human diseases that are equally, if not more difficult to tackle than the pandemic. Unlike COVID-19, major human maladies such as cardiovascular diseases, cancer, neurological disorders, diabetes are claiming lives from the young and the elder every day in every corner of the planet. This chart from CDC lists the top 10 causes of deaths in the US from both 2018 and 2019. Now for 2020, obviously, COVID-19 will mostly rank among the top 3, but we know that this ranking will probably most likely return to this order in the near future. So while early diagnosis, of course, will play a critical role in understanding and controlling these human diseases, but really treating the patients suffering from these disorders really call for the discovery and development of novel therapeutics and medicine. So this raises a question maybe in the audience that so how we have been doing at this front of new drug discovery in the past few decades. Here's actually a bar graph published a month ago summarizing the number of new drugs that has been approved by FDA each year, ranging from 1993 to 2020. Colored in blue are the biological drugs, which are proteins or RNA-based therapeutics. And colored in orange are the small molecule compound drugs that you would usually take as a pill or a tablet. And as you can see that from this chart, small molecule drugs remain to be the dominant form of new drugs, although there is definitely an increase in the number of biologics, particularly antibody-based drugs in recent years. And this really reflects the recent revolution in immunotherapy for multiple human diseases. You probably also noticed that at the turn of the millennium, year 2000, the number of new drugs and the started to rise. It's actually decreased or reach a bottom, and they only started to increase around year 2010. So interestingly, the number of new drugs was actually descending from 1995 to 2000 or 2002. And the government spending on basic research actually at this time was rising and almost doubled. So this actually reflects that the fact that it takes 10 to 15 years before the number of new therapeutics start to climb up. And that is the process of how long it takes for new therapeutics to be developed, starting from new ideas that are stemming from basic research. So here I'm stating something that's very important and obvious, that new ideas and new therapeutics really do not originate from pharmaceutical companies, but they come from curiosity-driven basic research carried out in individual laboratories at research institutes and universities. So in the rest of my talk, I will give you a perfect example where I think that we learned firsthand in our lab, where we were at the very forefront of identifying a new direction in drug discovery. And we believe this new direction is not just relevant to a single disease or a single drug target, but really is amazingly applicable to many targets in a variety of human disorders with wide sweeping significance and potential. So to better understand the story, let me first describe how conventional drug works. Okay, so if you know someone who have high blood pressure or arrhythmia, this person most likely will be taking some antihypertensive drugs, okay, to control the hypertension. Now, one class of these drugs, here exemplified as a drug called verapamil, actually target a class of proteins called voltage-gated calcium channels. These channels prolong action potential and drives the cardiac muscle contraction. So how does this drug actually help lower blood pressure? Well, it essentially actually plug the central pore of this iron channel and block the permeation of calcium iron. So it binds to the center of the protein and physically block the pathway that the iron has to go through, okay? This is one example. A second example would be a drug called Gleevec. And this drug was named Magic Bullet by Times Magazine because for its treatment for this blood cancer called chronic myeloid leukemia. The way this drug works is to insert itself at the active site of its target, which is a kinase, and the kinase abnormal activity would drive uncontrolled cancer cell proliferation. So I hope you can appreciate that in both cases, these small molecule drugs are embedded in the central pocket or cavity of their target protein, thereby blocking the action of these proteins. And these target proteins, therefore, are called druggable targets, conventionally speaking, because they do have a pocket that can accommodate a small molecule that acts as a drug, and this pocket is actually functionally important for the protein. By either blocking or occupying this pocket, these drugs would block the action of these proteins, and that leads to clinical efficacy. Now, interestingly, if you look at the human genome, which encodes roughly 30,000 proteins or 30,000 genes, they actually, these gene products do various things in human cells, but only 1/10 of these proteins, that's around roughly 3,000, are considered druggable proteins. That these proteins have a pocket, and these pockets are functionally important. You can use small molecule to block. The rest of the 90% of human genome do not have any functional pocket, and they are conventionally considered as undruggable, thereby intractable to conventional drug discovery approaches. So unfortunately, malfunction or dysregulation of some of these 90% undruggable proteins actually play a critical role in driving human diseases. Here are two examples. In cancer cells, we know that a very well-known oncogene called MYC is overexpressed and amplified. It binds to DNA and drives gene expression. The majority of this protein is in the disordered form that's represented by this dotted line. And this protein does not have a defined pocket for a small molecule drug to bind or to inhibit its function. Under Alzheimer's disease, which is one of the top 6 leading cause of death in the US, a major disease-promoting protein called tau, okay? It's also a very flexible protein. It does not have a defined structure, and it tends to aggregate into neurofibrillary tangles, and that is toxic to the neuron and cause or accelerate the development progression of Alzheimer's disease. And this is also considered as a classic undruggable target because it does not have any cavity or pocket for small molecule to bind. So then how do you go about these well-known or well-validated disease-causing protein targets? How can you downregulate their activity, thereby elicit clinical efficacy? And this remains to be a burning question in the past few decades that remain to be answered. Now, it's interesting that our research actually led to perhaps an answer to this question. And this idea or the story come from a rather unexpected field that seems to be completely unrelated to human biology. And they come from the green kingdom of life on this planet, that's plants. So we know that our ancestors extract medicine from plants, and many of which are still clinically used today. But here we actually extract not a medicine, a compound, but a principle or an idea, if you will, from these lovely living plants, organism that support the entire ecosystem on Earth. And remarkably, the whole thing was not actually started by us, but instead the topic can be traced back to an iconic science giant, no one else but Charles Darwin. So we know that Darwin for his evolution theory, but not many people know that Darwin actually published many books on a variety of topics in biology, including this one entitled Power of Movement in Plants, which was published in 1880. That's 20 years after the publication of Origin of Species, but 2 years before he died. Now, in this book, Darwin studied the slow movement of plants, which are widely considered as sessile organisms that do not move. Okay, so highlighted by his studies of finch on Galapagos Island, you might know, he was actually very fond of birds, and he always placed some grass seeds on his ceiling, so windowsill every day to feed the flying animal. Now, while most of the grass seeds got eaten by the birds, some of them actually start to germinate and grow into seedlings, standing on the windowsill. And not only that, these seedlings, as you can imagine, would bend toward light coming from the outside of the window, a process now known as phototropism. So Darwin actually became curious about this process and started to spend hours and days to meticulously trace the slow bending of plant seedling in the dark room toward the candlelight. And here's actually figure 181 from this book. Whoops. From this book, where he described this process by tracing the plant seedlings' slow movement. Now, we are, of course, in 21st century. We don't have patience to do this type of careful research. But here is actually a YouTube version of what Darwin saw back in 1880. And this is a time-lapse video of corn seedling bending toward blue light shining from the right side. And of course, this is a fast forward, rapid depiction of the process, which is otherwise very, very slow. So what Darwin noticed was that the bending is always triggered by the tip of the plant seedling, and by making a small cap out of aluminum foil to cover the tip, he was able to abolish this whole process. So he actually wrote at the end of the book by stating that we must therefore conclude that when seedlings are freely exposed to a lateral light, some influence, he called influence, is transmitted from the upper to the lower part, causing the latter to bend. Okay, so this Darwin's influence, at that time, he had no idea what's the nature of this influence, later was found to be a simple chemical called indole-3 acetic acid, which actually functioned as a master plant hormone controlling almost every aspect of plant growth and development. So this is a pivotal plant hormone dictating many biological processes in plants. And long story short, many decades studies after Darwin's time indicated that actually this plant hormone, this naturally occurring small molecule compound, actually induce specific protein degradation when it's generated by one part of the plant that's exposed to light. And in doing so, it actually promote transcription or gene expression in plants, thereby promoting the growth of the plant cells on the shade side. Okay, and even what's more remarkable was it was not until 2006 did we truly understand that this plant hormone actually targets a class of proteins called E3 ligases in plants that actually label whatever protein that binds to it with a marker called ubiquitin that marks the protein or earmark the protein for destruction by a cellular organelle called proteasome. It's a protein destruction complex. So basically, Mother Nature has evolved through billions of years of evolution, a small molecule that can control biological processes by actually inducing the degradation of certain proteins that applies as a brake for cell growth. In this case, by degrading the braking molecule, you can actually promote phototropism in plants, and of course, many other biological processes in these living organisms. So, very interesting enough, we actually was fortunate enough to learn about this story and used a method called X-ray crystallography to determine the atomic structure of this entire oxygen perception system back in 2007. Okay, we were wondering about how this small molecule that's evolved in nature can control this process of protein degradation with very high specificity. And obviously, I don't have time to actually go through the details, but really what's surprising was that we found that this plant hormone does not act as most of the known compounds that either acts as animal hormone or as drugs, meaning that even though they bind to a protein pocket, but they don't actually change the conformation or the structure or the shape of the target protein. What it does, it actually binds to a pocket that's actually represent the gap between two proteins, the ubiquitin ligase and the repressor protein. Okay, and in doing so, it acts as a molecular glue that would allow protein-protein interaction to happen at a very high affinity. So in other words, this small molecule really is an interfacial molecule that promotes protein-protein interaction. Now, this is a terribly interesting because protein degradation leads to the degradation of these repressor proteins or elimination of these repressor proteins in plants. And these proteins are actually considered as undruggable proteins because they themselves do not have a pocket. The pocket actually is sitting on the side of this so-called E3 ligase enzymes, which actually promote the degradation of whatever binds to them, these repressor proteins. So here, plant biology research taught us that there's a possibility that you can develop small molecules that could actually downregulate or eliminate a target protein that's non-druggable by promoting its degradation. And these small molecules can actually act as a molecular glue to resurface these E3 ligases to achieve this goal. Now, we know this happened in plant cells. Does it happen in human cells? And that was a burning question that remained unanswered for quite a while until very recently that in recent years, we got a positive answer to this question. And this actually leads to another story that I need to tell you, coming from probably most notorious drug in human biomedical history. And this is the drug called thalidomide. And many of you might have heard of this drug, which caused birth defects in Europe. So this is a drug first marketed in late 50s in West Germany, never reached the United States. It was it was marketed as an over-the-counter drug, and it can be cured. It can actually has efficacy against anxiety, insomnia, and other medical conditions. And it was actually widely used by pregnant women to treat morning sickness. And as soon as this drug appeared in West Germany on market, actually it caused what we call actually an epidemic of a birth defect. Okay, so turns out that the marketing of thalidomide triggered the outbreak of birth deformation because of its what's called teratogenic effect, basically causing babies to be born with birth defects, particularly characterized by malformation of limbs and other developmental defects. Now, despite the quick withdrawal from the market, this strategy actually was long-lasting, and still there are many disabled patients alive these days. More than 10,000 of these patients suffered from this notorious drug. The thalidomide strategy did not reach USA because thanks to FDA, which actually blocked its usage in the market. Now, with this, you probably will be surprised that this drug later was repurposed in late 90s, and it's actually used as a very effective drug treating the second most prevalent blood cancer called multiple myeloma. So this thalidomide drug and two of its derivatives called lenalidomide and pomalidomide becomes the best-selling small molecule anti-cancer drug in the United States and in the world. So interestingly, as long as this drug is not taken by pregnant women, it is a rather wonder drug, and it can treat blood cancer and a number of other diseases. Now, what was puzzling over the years, over the decades, was that how this drug works. Nobody knew. Actually, nobody knew the molecular target of thalidomide. So a number of studies, particularly starting around year 2013, led to a groundbreaking discovery that these molecules, these drugs, do not act as conventional drugs, but they actually work just like auxin. They promote the degradation of certain proteins, transcription factors, and kinases. Some of these proteins actually are considered undruggable targets. And structural studies actually shown that the mechanism of action of IMiD compounds, or thalidomide or lenalidomide or pomalidomide, actually recapitulate what auxin does in plants. So on the left hand, you have this naturally occurring small molecule acting as a molecular glue at the protein-protein interface to promote protein-protein interaction and leading to degradation of the target protein. On the right, you have a clinically used drug, which was developed without knowing its mechanism of action, doing exactly the same thing by resurfacing the human E3 ligases so that it can bind and promote the degradation of target proteins. So this actually really raised the or start the race of prospective development of molecular glue-based protein degraders. And because this really is a new direction that's approved by naturally occurring small molecule and approved by a clinically used drug, but it raises a question of how to actually develop this prospectively, meaning how can we intentionally look for it? Both auxin and thalidomide are retrospectively understood, right? How they work. When thalidomide was first developed, nobody knew how it acts and how it what molecule it targets to. Now we understand its mechanism of action, but can we actually use the same principle to prospectively develop drugs targeting undruggable targets? So this is what our lab is most excited about these days. We are actually pursuing 3 holy grail targets that are all conventionally considered as non-druggable targets. On the left side, you have KRAS, which is holy grail oncogene mutated in many type of human cancers, and particularly in pancreatic cancer. 98% of pancreatic cancer carries KRAS mutation, which causes abnormal activation. And so far, after many decades of research, very few people come up with, or team come up with any inhibitors that can effectively tame this oncogene. And we are trying to actually work with a small pharmaceutical company called Beyond Spring, partnership with Eli Lilly, to develop molecular glue drugs that can actually induce the degradation of KRAS through specific human E3 ligases. At the same time, we're pursuing MYC, the oncogene I highlighted before, through a startup company called Coho Therapeutics, using the same principle. But perhaps equally exciting is that within our own lab at the University of Washington, we are pursuing the tau protein that I highlighted, which underlines Alzheimer's disease. In Alzheimer's disease field, pretty much the more than 200 clinical trials has failed for pursuing another target in this pathway, beta amyloid, and now the focus is on tau, and yet tau is an undruggable target. How can you get rid of tau? It's a burning question. We think we might hold the key by developing molecular glue compound that can induce degradation of tau, thereby clear this toxic proteins. Now, I don't have time to actually really tell you the details of these ongoing efforts, but I have to say that I want to say that it's actually what we're doing is turned out to be quite promising. So to summarize, unmet biomedical needs in the 21st century really calls for new ideas, new notions in drug development. And because the majority of the human genome is considered undruggable, we really need a new approach to take in order to eliminate or downregulate widely validated undruggable targets. And we learned a very important lesson from plant hormone, auxin, which actually acts through protein degradation. And this is echoed by a clinically used drug, thalidomide, and its analog, collectively known as IMiD. And we think actually targeted protein degradation, exemplified by the naturally occurring small molecule and the clinically used drug IMiD, really represents a new direction for us to drug the undruggables and hopefully to be able to come up with new modalities in novel therapeutic drugs. So in this summary slide, I really want to finally highlight the fact that basic research is extremely important, and curiosity-driven science really leads to the new ideas that otherwise will not come out of void. So by that, I want to conclude my talk and acknowledge all lab members in the Zheng lab. In the past 20 years, I'm really fortunate to work with many talented young people. Many of them actually come from Asia, come from China, who work with us and work with each other to enable us to make new findings. And we are, of course, thankful to the collaboration from numerous groups around the nation and around the world. And we're funded by National Cancer Institute, National Institute of Health, and also Howard Hughes Medical Institute. With that, I conclude my talk and welcome any comments and discussion after Dr. Liu's talk. Thank you.